button. So if you just listen, you don't have to hear the whole recording, but if you just listen to the first few minutes, you would be hearing the questions and hearing the answers, okay? So during your, if you wanted to do it that way. But okay, so let's go ahead and go over this lecture quiz, making sure that it was fair and the questions were fair and that you feel pretty good about these. Are these from today? Sorry. Do I have them all now? I have them all now before I start going over them? Okay. So, are we good? Everybody's ready to listen up? Okay, so the first question said to write the equation for aerobic respiration. I thought that was fair because I told you all it was one of the few you would need to know. You need to know it. So what you wrote was exactly C6H12O6 plus 6O2, an arrow moving to the right, and then you wrote 6CO2 plus 6H2O. If you did not know that for today, that's fine. But what you need to know is know it before this first test, because are you likely to see all of these again in a multiple choice format? Yes. Yes. Plus other things that I didn't ask you about, but anything from lecture. Right? So if you miss something on these quizzes, you don't get too bent up, but you make sure you get it going forward, right? So list the 4 major categories of organic molecules that make up all living organisms. You said carbohydrates, lipids, proteins, and nucleic acids. Right. Define pH. It was fine for you to put the negative log of the concentration, which is brackets, of the hydrogen ion. So you wrote a little positive about the hydrogen. And maybe you just said it means that you understand how many hydrogen ions are present. Maybe you wrote that, and that's okay too. All right. You're going to have to know everything we talked about with pH for a multiple choice test. I gave a lot more than just that basic definition, didn't I? So you might have multiple choice questions related to that information I gave you, and that would be fair because I definitely talked about it extensively. What is the normal range for most of our fluids in our bodies? The range is 7.35 to 7.45. So that's what you put. Which category of organic molecules do our cells most depend on for clean, quick sources of fuel? What is that? Carbohydrates. What 2 types of human cells exclusively depend on them? Nerve cells and red blood cells. This was one of the issues I know and I don't know who it was, but somebody had wrong in your study guide. So anyway, make sure you understand all of our cells need glucose. That's our best, cleanest, quickest source of fuel. But a couple of them must have. So that's very different. That's very different than the way that somebody had in their study guide. Explain what is meant by the primary structure of a protein. So what I wanted you to do in a few words is just say that this is understanding which of the 20 amino acids, which amino acid is in which spot on the chain. That's what I wanted you to say, period. Okay. And I might have had that worded in a lot of different ways and you know, could have 28, 9, 30, how many people are in here, different ways that was said, and it may all be right. What is the function of a buffer? I simply wanted you to say buffers help to resist pH changes. Or you could have said something like a buffer is going to keep pH in its normal range. A buffer is going to help to do that. Okay. Destructive molecules known as blank can be produced in both normal metabolism but in higher numbers by exposure to harmful environmental influences. So what is that thing called? Free radicals. Again, somebody on the study guide had that as a wrong thing. But anyway, these molecules can be eliminated with what? What produces antioxidants? Our cells. Our cells produce antioxidants just to deal with free radicals. And so antioxidants neutralize free radicals. You got 20 points for being here. And I do appreciate you all being here. And then I asked you to tell me what exactly ATP stands for. I think I had asked you to know that. And it is adenosine triphosphate, right? I didn't ask for the function of it. I didn't ask you to draw it, which you have to do in organic chemistry. But no, anyway, so you just, yeah, organic chemistry. If you ever get to take organic chemistry, well, anyway, you will be drawing all those molecules. But anyway, I just asked you to tell me what it stood for, adenosine triphosphate. All right, are these fair questions from lecture? Were these fair? Were these questions that you felt pretty good about? Okay, all right, again, these lectures are to serve some functions. They're to serve functions to encourage you to stay up to date with the information. Because if I were to get you quizzes and all of a sudden you had a test on chapters 1 through 4, I have about 20% of y'all passing this class. And that's a fact. But because you're sort of made to stay up, so that's one function. The other function, though, is to also let you know how well you are doing. You know, and it sort of gives you an idea about what you might need to do to change up if you're struggling. Okay, and we're all in this together. And so I have some of you that I know right now, wouldn't hurt everybody, but need to be in that learning resource center for tutoring, right? So because it can help, every resource available can help, and I hope you'll take advantage of that. But you know, we're adults, and so you figure out what you need to do to get through. The scientific measurements that we just did. There's some people who just struggle a little bit with math, and especially if you're being tested on it. I don't want people to worry too much. If you didn't do great on that, we're going to have lots of lab quizzes. It'll be all right, right? But again, this is something that you do. This is the basics that you have to do drug dosage. So if you didn't do great on this, it's not a big deal at all, but work on it, right? So just work on it and make sure you get it. Fair enough? Any questions for me?
What is that first chapter test?
Oh, it's, I don't know.
Okay.
Okay. It's going to be, I'll give at least a week's notice that a test is coming. And I don't know if we're going to do 3 chapters. We're about to start our 3rd chapter. And after I do this chapter, I might think, you know, they've just had enough. Let's go ahead and do a test, you know, and get that out of the way and do that. So it probably really will be the first 3 chapters. So that means that pretty soon, probably, you know, maybe not next Thursday, but the Tuesday after, you know. And it'll be multiple choice questions that will be from the lecture, also from deeper insights. Does everybody know what I mean when I say deeper insights? I've gone over that in the class. Okay, so you are supposed to be reading those deeper insights and taking little notes. And here's the thing, I had a student very recently tell me this, and I thought that's brilliant, which I thought of that. Since your final exam is comprehensive, back to chapter 1. So your final exam is going to be 16 chapters of information, but every single question comes from the deeper insights. And on chapter 6, we'll start with the chapters will have a disorders table at the end of them. The organ systems have, once we get into organ systems, there's going to be this little table, usually half a page, that talks about disorders of that organ system. So every single question on the final exam will either be from a deeper insight or a disorders table. So I had the student a few semesters ago tell me that they kept those notes separate in a little book. And they studied their deeper insights, and when the disorders table started in chapter 6, they studied those for the test, because you have to for each test. But then that's all they pulled out and studied for the final exam, and they had them all together. And I thought that was genius. So anyway, don't know why I hadn't thought of that. But y'all, that's just a little hint for you. So you might be wanting to keep like a little separate folder for your notes on your deeper insights and your disorders table. Okay. If I have a question on a test that wasn't from lectures or the deeper insights and eventually the disorders, I throw that question out. So do you all know how to study? Do you know what to study? So please use that, okay? All right. So did we finish chapter 2? Did we?
Yes.
Okay, we did. I thought we did, and then I had a moment panic this morning thinking, no, we didn't finish chapter 2, but I thought we did, so great. And I can ask you any questions about DNA that y'all know what DNA stands for, deoxyribonucleic acid or RNA, but I gave you some basic information, some introduction to it, then the molecules. And the reason I do that is because we're going to expand on that when we get to the next chapter 4 is all about genetics and inheritance and heredity. So the reason you have things, the reason things are presented to you in the format that they are is that you need to own them before we get to the next. And if you do, it makes the next easier that we get to. It's the same reason that your lab 2, you're looking at some medical terminology, some directional terms, aren't you? And regions of the body. Because when we get to bones, if you don't do well on lab 2, I see you suffer with bones. And if you don't do well on lab 2 and bones, I see you fail muscles and not know how to label vessels when we get there. And you just struggle the whole way through. So let me say this to you. You're being presented this information because it's logical, and you have to own the foundation to be able to have a strong understanding of what's coming next. So that's my encouragement to you, to make sure that you are getting this. You don't have to have it by the time I quiz you on it, but you should have it soon after, okay, for your sake, all right, before we get to this next information. So that's my encouragement for you all, to stay present, stay focused, and stay up. It's a lot easier to stay up than to catch up. Okay, so that's my encouragement to you. Can you tell I was a cheerleader back in the day? I hated it. But no, I really did. But anyway, I was, and so anyway. So here we go. We are looking at cells, and you all, I've already given you a definition. You understand that the studyology of cyte, C-Y-T, Latin for cell, this is what we're about to look at now. And we already know some things about the cell. We absolutely know that Louis Pasteur finally did an experiment that put that theory of spontaneous generation to rest. And we now know that cells are the smallest things that have properties of life. And we also know that all life comes from pre-existing cellular life. So be reminded of that, because I'll ask you again. It wouldn't make any sense that our human cells all look the same under a microscope. It takes a microscope to be able to see them, because we cannot see them macroscopically. Macro means they're large and we can see them with our eyes. It takes magnification, doesn't it, to see these cells? And we're about to start looking using microscopes in the lab. That will be your third lab quiz. I haven't even given you your second one. I'll talk about the third one. Lord have mercy. But anyway, that's our next thing, because we're going to start looking at these human cells, and we need to understand it wouldn't make any sense that they look the same, because there's over 200 different ones and they have different functions, and structure dictates functions. So these are some of the descriptors that we use for cell shapes and sizes. And if you looked at some of these pictures, how many of you are visual learners and you like pictures? Me too. I'm a visual learner. I like graphics. I like pictures. I like colors. Colors help me to know things and remember things if I'm highlighting with different colors. So figure out what you need to do to get this, but definitely get this. Squamous or squamous, both are accepted pronunciations, but squamous cells are thin, flat, kind of irregular. They're really amorphous. And when you talk about morphology, that's the study of shape. Something being amorphous, a or ana in front of the word means what? Without, right? Without. So amorphous means that they really don't have too much of a shape. They're going to be influenced by whatever the environment is. So sometimes they look really thin, sort of flat, sort of amorphous, right? Poly means what? Always means what as a prefix? Multiple or many. So polygonal, these are going to be irregular shapes with many sides, with many kind of sides. Stellate is star-like. And again, we can see pictures in a second, but cuboidal cubes, y'all know to be what? Squarish, right? remember, we'll be looking at these cells into our eyes sometimes on these illustrations, and certainly even looking through a microscope, they appear to be flat, but are cells flat? Obviously not. They're three, or we'd be flat. They're three-dimensional, and so we know that. Spheroidal, or columnar is the next one. Columns, shaped like columns. Spheroidal, obviously y'all kind of know that word, and disc, looking like a disc shape. So they're described based on just their anatomy and what they look like. Fusiform is going to be kind of thick in the middle and tapered at the ends. That's what that means. And fibrous is sort of thread-like or wormy-like. So again, when we start our study of microscopy, when we start with the microscopes and we start looking at the microscopes at these cells, we're going to not expect them to all look like little cubes, are they? That wouldn't even make sense to us. So we're going to see them, and we're going to see that they have these different shapes. Have a basic idea about what those descriptors are, what they mean. And most of them, y'all have heard terminology like that: columnar, cuboidal, spheroid. Y'all have heard that, but have an idea about them. Would they be all the same size? No, of course not. So some are much tinier than others. Some actually have really kind of appendages that make them more than a meter long. Isn't that crazy to think that you have cells that are microscopic, but they have these kind of appendages that go out that make them about a meter long? And those would be our nerve cells. So anyway, of course they don't have all the same. What dictates whether a cell is going to be disc-shaped like your red blood cells or stellate like your nerve cells are going to be? What dictates that? What shape the cell is going to take? The DNA of the cell. We said the DNA of the cell. You all started as one cell. But wherever the DNA opens up, if that cell becomes a nerve cell and it's going to be stellate with a long appendage off of it, it's because the DNA opened up there for those recipes. Where if it becomes a red cell, that DNA opened up in a different place. And it dictates. It also dictates, did I lose your place? Okay, it also dictates where, like how long the cell will live. Do you think all cells live the same amount of time? You have some cells that will only live for a few hours in your system, and then you have other cells that need to live for decades. So those cells that need to live for decades, like your central nervous system tissue, your spinal cord and brain, do we want anything damaging them? No, because guess what? You don't get them back. They don't regenerate. You don't get them back. But some cells can, and some cells only live for hours. Again, what dictates that, Andrea? It's the DNA. The DNA dictates everything that's going on inside the cell, what the cell shape's going to be, what kind of proteins it's going to make, what its functions are going to be, and even how long it's going to divide and or live, right? So I hope that when we've been talking about this kind of makes sense to you a little bit. Now, here's what I want you to know about a microscope. And we're going to do our microscopy on microscope stuff in the lab. And actually we will, by the way, we will start that Tuesday in lab. And actually, Tuesday, y'all remember you have a lab quiz? Number 2 lab quiz, right? You have a lecture quiz from today at the beginning of lecture, but at the beginning of lab, you will also have your lab 2 lab quiz. Any questions about what's going to be on that lab quiz? Feel pretty good? Yes, Tiara.
Is it just the medical terminology and
Medical terminology, directional terms. There were a couple of things on the visible body, right? And then that page. Was it page 30? I think. Yeah, it's that page that has the anterior and posterior aspects of a male and female with different regions, right? So y'all know that that's where that lab 2 filling in the blank questions are coming from. Anything that's on those sites should be fair game. You've had them now for quite a bit of time, by Tuesday even more. So manage your time. Time management is the biggest thing in our lives, isn't it? So manage your time well, that you have gotten to really be very familiar with that by Tuesday. Fair? And we're moving right along. So, okay, and Tuesday in lab, we will actually after that lab quiz, we'll get the microscopes out and we'll start our study with microscopy. We will be answering questions and labeling microscopes. So this is just FYI for your lecture part, okay? I'm not asking you anything in here about that. Now, this picture, if you will, of a cell that's actually beside another cell and another cell. This illustration of this type of cell, it almost looks like a columnar cell, doesn't it? It looks like a column. Can y'all see that? It's kind of like a column. And it's been sliced so that you can see inside of it, and it looks like there's all these components inside of the cell. We already have learned about what some of these are. We know that cells are composed of organelles. Didn't we learn that? And we had some examples of names of organelles. What were they? Mitochondria, nucleus, Golgi, or Golgi, whatever you want to say. And Golgi apparatus, we said, I don't know what else we said. Did we name any others at that time? But anyway, so y'all had learned that. So y'all knew there were going to be these things inside of cells. So in this chapter, what we're going to do, and this is your goal, is any of these organelles or structures that I talk about, you all will know its function. You could match functions to structures, because that's your goal, forget it, from this whole chapter really is essentially that. Fair? All right, now look, I meant to ask today, this makes me crazy, it makes me remember. I meant to ask you all today, what is the function of phospholipids? And what would you have said? They make up most of the cell membrane, plasma. So y'all, do you think you're going to have that on first test? I can promise you. So I'm glad you knew it. Genetic, right? I have two genetics. Spelled different, but two genetics. Exactly. So that's not going to go. That study's not going to go to waste. Believe me. It's just that I didn't get to ask it as one of the 10 questions. All right.
So we'll just need to know the function. We won't have to label this.
Yeah. That will be a lab. Okay. That would be in lab, wouldn't it? Yeah. And so we will actually be looking at all of these, looking at their shapes and structures, and seeing how their shape and structure of even the organelles helps us kind of understand what they're doing, because it does. All right. So when we think about basic things of a cell, when we think about the basic things of a cell, we understand that this little membrane is a bilayer of phospholipids, and it's a bilayer, and this membrane is keeping the outside out and the inside in. So it forms the boundaries of the cell, doesn't it? And it gives the integrity to the cell, if you will, strength or integrity of the cell. So that's what it's actually doing. And inside of the cell, when we see plasm, this means fluid. So it's the fluid part of the cell. These cells are filled with fluid. You said about 65% of your water is inside of your cells, didn't you say? So that means the cells are filled with fluid, all right? So we think about this with the cytoplasm. We will also, in a little bit, talk about something called the cytoskeleton. Actually, let's go ahead and talk about it now, but I think there's better pictures of it later. But the cytoskeleton is made of proteins. And you know what I think about with this, and it helps to understand the functions? It's the cytoskeleton, and they're not really showing it to you on this illustration, but it's almost like if you looked at, have you ever gone out early in the morning and you see, it's like spider webs in the dew, right? Well, inside of your cells, you have all of these proteins that are crisscrossing, almost like an intense spider webby look. And what those proteins are going to be really the skeleton of your cell, the highway system of your cell. They do give structure to the cell, but more importantly, they are the highway system of the cell. So things that are being produced here and made down here can be shipped and sent to other parts of the cell. Guys, you can think about, like the United States has a highway system. So how many of you have eaten an orange in the last month? Only got two. Okay. Well, okay. But okay, you didn't get it from Hazel. Where'd you get it from? Florida. You got it from California, Florida. You got it somewhere else. How did it get here? Y'all can think about that as the same way as inside of a cell, because if something's being produced here but needed up here, how's it going to get there? Through the what? Cytoskeleton. This is the highway system of the cell. it's referred to as the scaffolding of the cell, and everybody knows what scaffolds look like, right? And it really is that. It's support, it's structure, it's how things are transported, it's how things are moved. And the predominant component of it is what organic molecule? Proteins. All right. The predominant component of plasma membranes is what? Lipids. Facts, right? Lipids, right? Lipids. Proteins play a huge role too, and we'll talk about that in a minute, but whatever. And then the extracellular fluid, extra means the fluid that's outside the cell. And even when cells, certainly on this top, apical means just on the top surface, certainly on the top surface, you know, there's a visible, you know, looks like there's nothing there, but that means there's fluid there, right? But even between cells, do you think there's got to be a fluid interface? There really is. Even between cells that are very tightly close together, there is definitely a fluid interface too. So there's this extracellular fluid we talk about too. All right, so here you go. Here's some good pictures of cells that they try to give you this three-dimensional aspect of it. So you've got the cell here, understanding there's tons of things inside the cell, and then they've taken out a wedge, haven't they? So you can kind of see. And there's all kinds of illustrations that you can find. And this might seem a little crazy to you at this point, but we're going to talk about all these structures, and it's going to be just second nature for you to go and say, oh yeah, I get, you know, I get that that's the nucleus, and I get the mitochondria, and I see the Golgi. They always look the same, no matter what size cell you're looking at. The organelles don't change shapes depending on the shape of the cell. The organelles keep the same basic shapes. So let's take a look at the plasma membrane first. You ready? And if I were to take, and by the way, this is if I took a cell right here, and I took a slither. I just took a tiny little slither of the plasma membrane. This is what I'm seeing right here. This is the extracellular component. This is the intracellular fluid. Are y'all with me? And I am seeing just a tiny, tiny little bit of the plasma membrane. And what does it look like to you? Even from back in the back of the room, can you see that it's a bilayer? Can you see that? And y'all see that it's a bilayer, and you know it's a bilayer of phospholipids, keeping the outside out and the inside in. Now, this is the intracellular fluid, and this looks like a membrane-bound something right here as well, doesn't it? All right, now look, so let me tell you something. This obviously is an illustration. Beautiful artistry. I maybe even have that on my wall. Not really. But this is an actual human cell that has been sliced and looked at. This is called an electron micrograph. We can't see this kind of detail with our regular clinical grade microscopes, light microscopes, but with electron microscopes, we can see 100 times more detail. And this is a detail of an actual human cell. Okay, now with those scopes and being able to see this kind of detail, does the anatomy talk to us about what they must be doing with these structures? And that's how we have learned, that's part of how we have learned what these organelles I'm about to tell you do. Many people say they're so glad when we finish chemistry and cytology, these first two chapters, chapter 2 and chapter 3. They say they're glad because, you know, that this stuff is out there for it. No, it's not. No, it's not. It's just that we can't see them with our eyes, but we can see them with electron microscopes. These are real. Okay, so we can see this. Use the pictures. Use the technology. So is everybody good? Everybody's good. Nobody would have electronic devices out in here because I hate to drop people from class. Okay, so good. Now we're going to take a look at this plasma membrane and see how it functions the way it does. Would it make sense to you? Would this make sense to you that the cells need to get some things in and need to allow other things to go out? Would that make sense that it can't just be a barrier and like a wall? We won't talk about walls, but okay. It can't just be a barrier and a wall. It can't. It's got, the definition of life is that cells can breathe things in from their environment, change them into a usable source of energy, and do work. And then also that your cells are talking to other cells. That means they've got to secrete products from them. And all of our cells do. So how does a plasma membrane do that? What you're seeing right now is an illustration of just the top part of a plasma membrane. And you're seeing the little you're seeing the phospholipids, the hydrophilic heads. Do you see this? Because we said our cells are bathed in what? Water, and we know that there's hydrophilic heads to the inside of our cell with the tails pointing to each other. So that's what you're seeing. But there's a ton of things embedded in that. And what these things predominantly are, they're not exclusively, but predominantly they're proteins. Proteins that have very specific functions. The plasma membrane has the function of just forming the barrier. But these proteins that are embedded in the plasma membrane, they're going to serve the function of actually helping the membrane allow for certain things to come in and certain things to go out. This is called selectively permeable. Have you ever heard that cell membranes have selective permeability? You know what that's a fancy way of saying? They let some things in and they let some things out. That's a fancy way of saying, right? So anyway, and how do they do it? By embedding particular types of what? Proteins that have very differing functions. Now let me give you some examples of these proteins. There's also some carbohydrates and whatever. So, but mostly I want you to think about proteins. So let me say this, what some of them do. Some of them act as doorways that act very specifically to allow specific things in. For example, some of them will only recognize insulin. And when they see insulin, what will they do? They will open the doorway and allow insulin to escort what into the cell? Glucose. Exactly. Some of them are going to have a very specific shape. What dictates protein shapes? DNA. By dictating the primary structure, which is going to then help us to understand why it coiled or folded, or do you get what I'm saying? Did you just have a aha moment? Oh, we recognize aha moments here. That's so awesome. Yes.
Is that like when you were talking about receptor proteins?
Yes, that's exactly what I was talking about. Receptor protein. Some of them will recognize like cholesterol. Okay, so if it's, and by the way, is cholesterol a bad thing or a good thing? It's a good thing, but we can make most of what we need, so we just don't need to like flood our system with it, right? But it's a good thing, and we do have to, our cells need to have cholesterol in it. So some receptors will recognize cholesterol, and you bring it into the cell, be able to use it the way you need it in the cell, and that's going to be a great thing. I want y'all to write down familial, and I think I already told you this, cholesterolemia, did I? Familial, so I've already introduced that. And those people, their DNA lacks the recipe to make those receptor proteins that recognize cholesterol. So instead of cholesterol being able to come into the cells and get out of the bloodstream, it's staying where? In the bloodstream. And so is that going to be good for the blood vessels? No, it's not. And so we get that. We get that these receptors. So we have to have our DNA having all the recipes it needs to make all the protein receptors we need. We also have little proteins that act like flags. Have you ever given directions for your home to say, I'm that house that has the blue flag, you know, and that's your house, right? It recognizes you. Yes, Chris? I know you didn't ask me that. So it's just because, Chris, that's why we have our textbooks. You go to those. I can't really stand up here and spell, can I? I mean, I could, but would it take a lot of time away from people? So every now and then I will spell something for you because I don't think it's in your textbook, or it might be something that you can find. But I would just to let you know, you know, spelling is elementary. Spend the time to get it, okay? But it's elementary. You can look it up. So then you have these receptors that act as recognition proteins. So these recognition proteins tells your immune system cells, the cells that protect you, your police, your military, that this is you. This is your cell. So these, and I've just pointed to this one like it's that, but anyway. So every one of your cells have these self-recognition tissue receptor proteins that say this is you. And as long as that flag is out there, your immune cells are protecting it and are not doing anything to it. But Does anyone else have the same flags that you have? So if you get an organ from someone else or tissue from someone else, does that tissue, those cells that make up that tissue, have those same recognition proteins? So what will your immune cells want to do? Attack it. So anybody who gets any kind of transplant or any kind of organ donation, what must you give them forevermore? Something that is going to suppress their immune system, because otherwise they would do what to that tissue? Attack it, destroy it, reject it. Is that right? And what level is that on that's happening? A cellular, molecular level. Molecular with the proteins, cellular level, isn't it?
What's that thing called again? What is it called?
They're just special kind of proteins. They're actually called major histocompatibility proteins, but MHCs. But you know what? For now, just say they're recognition proteins. They're proteins. But if you, for some reason, hey, how about this happens? Because you're constantly putting your recognition proteins out there. Don't houses have to change flags? Don't houses have to change the roof? Don't you have to do that? Your cells are your houses. They're your homes. They're constantly having to change things. But what happens when it's influenced by a lot of things, usually environmental. But what happens when the recipes start to get tweaked a little bit and changed, and all of a sudden in an organ, these recognition proteins aren't there? What will your immune cells do to it? Start attacking it, start destroying it. Does anybody know what class of diseases that is? Autoimmune diseases. Auto means self. Immune system means that your immune system is attacking your own self cells. That's an autoimmune disease. That's exactly right. That all of a sudden, I have one. Y'all don't care about it, but just to tell you, this is a very common one. It's called Hashimoto's thyroiditis. 1 in 8 women over the age of 30 have this. But it's interesting. I was actually 30. I had no, so 30 years ago, had no symptoms or signs of it. It just got caught by error, really. And so I got caught before I ever had any signs and symptoms. So I was able to go ahead and go on therapy. But what happened was my immune system stopped recognizing my thyroid gland and started attacking it. So my thyroid gland stopped producing, couldn't produce anymore with the hormones that it needed. So now every day I have to replace the hormones that my thyroid gland should be producing, but it can't because it got destroyed by my immune system. You get what I mean? It's a very common autoimmune disease, but autoimmune means that your immune system is attacking yourself. So can you catch an autoimmune disease from someone? No, you can't. It's their own cells that are doing that. Name some other autoimmune diseases. MS, multiple sclerosis. What else? Lupus is an autoimmune disease. What else? Name some others.
Cystic Fibrosis?
No, that's an inherited disease.
I'm sorry. Would HIV be one?
No. HIV. Why is an HIV autoimmune disease? That's a virus. That's a virus that you can get from somebody. You can catch that virus that causes that disease. Some things are inherited diseases, but an autoimmune disease is when your own immune cells start to attack your own tissues. You cannot catch them from somebody else. Now, there is a familial component, but we don't think that it's autoimmune. So type 1 diabetes. Type 1 diabetes is autoimmune. Rheumatoid arthritis, which is very different from other arthritis diagnosis, but rheumatoid arthritis is autoimmune. There's tons of them, and we'll talk about them as we go. But those are ones you've heard of, aren't they? Aren't they all ones you've heard of? All right.
What about epilepsy?
No, that's not. Epilepsy is actually defined as a seizure activity that happens usually for unknown reasons. And so might it end up being considered to be something that's with the autoimmune system maybe, but right now it just means that you've ruled out every other reason, but you have epilepsy, seizure activity.
My daughter has it.
Pardon?
My 2-year-old daughter has it.
She
just found out.
Yeah, and they called it epilepsy. Okay. I'm sorry, because that's really hard. But the good news is, I mean, if you're looking for good news, the good news about childhood epilepsy is that the earlier they get diagnosed, the better potential you have of them outgrowing it. So if you stop the activity, the seizure activity, which is a learned activity in the brain, but if you can stop that long enough over months and years, you can usually wean off of medication and cure, really. Doesn't mean for sure that she will be, but hopefully that'll be her outcome. If epilepsy starts later in childhood or late as early adults, it's usually forever. But that age is a really good chance that she will well, it's not really outgrow, but essentially. Okay.
To better understand, the recognition proteins is the same as the receptor?
No, they're all different. They're all different from each other. They are all different from each other. Even the receptor proteins are different from each other. Some of them will receive like insulin. Some of them will receive cholesterol. You have the ability, your DNA has the recipes to make thousands of different recipes for proteins. And every one of them, because their recipes are different, their shapes are different, it means they have different what? Functions. Some of these surface proteins will act as zippers, if you will, so they can help to hold cells together. So these proteins that are embedded in your plasma membrane, do you need to understand they're there? Yes. Yeah, they do. You do need to understand their molecular weight's actually a lot higher than the phospholipid bilayer. But anyway, they're the ones that are helping the plasma membrane to carry out their functions. And those functions being selective permeability, or this way of letting some things in and some things out, not others. All right? All right. So this is an illustration, outside, inside, and you can see where these proteins are embedded, and these carbohydrates too chains play a role. Because we said that when we think about these organic molecules, they aren't just these divisions of these 4 divisions. Sometimes carbs, I told you, will play with proteins and make a big molecule, right? And sometimes carbs will play with lipids, and sometimes proteins will play with lipids. And so there's really usually more complexity than just saying they're these isolated 4 molecules, but that's just kind of FYI. And I would hope you all are all fascinated by what's happening at a cellular level for what is actually going on. Now, we said phospholipids 70+%, but actually when we think about the whole membrane, it's mostly lipids, isn't it? But do we have to have the proteins? Do we embed it? Yes, we do. Now, what else do I want to tell you about? I told you about the cholesterol, and one of the functions of the cholesterol, I think I had told you, the cholesterol acts as a stud in the walls. Isn't that right? You had a picture back there. I won't go back to it, but the little cholesterol molecules act as like the studs and give the integrity of the plasma membranes. I shouldn't say walls. I should say plasma membranes, because a membrane is very different from a cell wall. Animal cells don't have cell walls. They don't. Now, there are some proteins that act as what are called channel proteins, and I don't know which one they're, but they act like channels, and they're going to allow a quick movement, if you will, of certain products through, and others, you know, maybe not, but they're very specific, and they act like that. So what I want you to know is there's an entire family of diseases that result from these channel proteins. Because didn't we say all diseases is really at this molecular level? Right, right. So anyway, just so you realize that. Some are going to act as pumps and carriers. Let me give you an example of a disease that is going to be associated with one of these pumps. You ready? It's cystic fibrosis. Cystic fibrosis, have y'all heard of that? Anybody know anybody with cystic fibrosis? So you understand that the lifespan for most cystic fibrosis patients, they're lucky to get to 30. That's with modern medicine. Before modern medicine, they didn't get out of toddlerhood. This is the most common inherited inherited disorder of lighter tone people. Just like sickle cell disease is the most commonly inherited disorder of darker tone people, right? So what happens with in cystic fibrosis, and it's inherited it's inherited in the same path, the sort of mechanism. Don't say that if you inherit sickle cell, you have cystic. I'm not saying that. I'm saying the pattern of inheritance is the same as with sicklers. I talked to y'all about sicklers, didn't I?
Yes.
Okay, all right. So in cystic fibrosis, though, and we'll just do it with a capital F, if you have a recipe for chloride, your chloride pumps to function, you only need one recipe from one parent to have functioning chloride pumps. Okay, so anyway, I'm not sure. Oh, fibrosis is what I did. Okay, but you might have from the other parent got a variant of the recipe, and this is the recessive trait. But this is okay because does this person have cystic fibrosis? No. Does this person have fibrosis at all? No, they don't, because they have normal chloride pumps. They do. Now, if a person, though, has the genotype, meaning that they don't have a recipe from one parent and they don't have it from another parent, it means that their chloride pumps can't work efficiently. And these chloride pumps that can't work efficiently, what it means is they can't pump chloride out of the cell. And the reason that that's an important thing to do is because there's sodium out here. And because of that, you're going to get this nice saline layer. Because isn't there also water out here? So there's going to be this nice normal saline out here if you're pumping chloride out. And it's keeping this the surface of the cell bathed, right? But if you can't do that, and you can't get a nice saline layer out here, this can get sticky. And if this is getting sticky out here, can things be moved past the cell? No. So honestly, cystic fibrosis is it affects multiple organ systems, not just the respiratory system. It affects the reproductive systems, the GI, the nutrition. It affects every organ system. And it means that your chloride pumps don't have the right recipe to work the way they're supposed to. That's what it means. Do you have any questions about that? There's a deeper insight, I think, about cystic fibrosis. Certainly they go into more technical detail. But did I just break it down for you? The problem is their chloride pumps aren't working. And even with best medical care, there's different variants of genetic variants of cystic fibrosis. But for the major part of the disease, most people that fall into the major part of the disease, they're going to need lung transplants very early on in life because their lungs are going to be just devastated by the mucus buildup and repeated scarring from pneumonias. And so they're going to need lung transplants. And those lung transplants, if they last 5 to 10 years, they're usually happy, and they're going to need another one at the end of that. So it's a devastating disease. All right. And I told you about recognition or cell identity markers, didn't I? And I told you that sometimes proteins help the cells to stick together, don't they? Adhesion. All right, so the things I tell you about is what you're going to need to know. All right, now here's another thing that's kind of on the outside of our cells. It's on the outside of all of our cells. This is the glycocalyx. And glyce, we said, means what? Yes, sugar. So we think about carbohydrates. It's also got proteins and lipids. But anyway, so this calyx, this formation, if you will. You know what I call this? I call it the glycocalyx, and they don't have a great picture of it, I don't think. But anyway, I call this glycocalyx is really, it says a fuzzy coat. It's your sugar coating on your cells. It's kind of your sugar lining of your cells, if you will, on the outside of your cells. But the important thing is that this is where you're going to find those identity markers are in here. And this is where people are going to look for the tissue. Have y'all ever heard that when somebody gets an organ transplant, they have to find a match right? That means that they're looking for the tissue identity markers to match up as closely as possible. There used to be about 7 that they were looking at, and if they could get even 4 or 5, they were happy. Were they going to find somebody that had all 7? Only if they have a what? And do most of us have an identical twin that we can go to for an organ? No. So if you don't have an identical twin, you just got to get as close as you can. There's no such thing as going to be a perfect match. But they do have guidelines for how many of those markers they need to match, right? They're not going to find a perfect match. That's why they're going to have to forevermore take immunosuppressive meds so that their immune system won't destroy that. What does that put them at risk for? It means that their immune system is now compromised, and so they're going to have to be very, very careful, because even what usually would cause a minor illness could take them out, right? Because they don't have the protection that they would typically have. So, okay, I just want you to know that's where the glycocalyx is, where most of those self-identity it is where they're going to be found. Now, microvilli, I want you to see the microvilli. And this picture that they've shown you they've illustrated is of a cell that doesn't exist. The reason it doesn't exist is because the cells that have these microvilli are not going to have cilia. Okay, so, but they're just showing you a cell that would have any kind of thing that might be present. So these microvilli, what they are, and are you going to need to know their function? Okay, what they are projections or protrusions of the cytoplasm of the cell surface. So where some cell surfaces are smooth, where a cell that has microvilli doesn't have a smooth top surface, does it? It has these areas that have these projections. Now, what I'd like you to see, and I don't know, see, that's not going to be right on that, but what I'd like you to see, and maybe you can see this, if I were looking at a real human cell, and we know this is 3-dimensional, so we know we're looking at a piece of it here, and it was smooth, I could measure that surface area and know how much surface area there was on that cell. And if something needed to be absorbed or excreted from the cell, I know it would have this much surface area chance at it, right? Is that right? But if all of a sudden there were these protrusions of it, of the cell surface, what happened to the cell surface measurement? Did it go up or down? It extended incredibly, almost like phenomenally, didn't it? So now instead of just this amount of chance for something to be absorbed or taken out, you've extended the surface area tremendously, and now you have these many chances more for absorption. So that's just a lot. That's just a lot of words to tell you that microvilli function, and the cells that have microvilli, they function by rapid absorption or usually absorption. So rapid absorption. Does that make sense? Does it? Okay. So the function is for rapid absorption. Do you need microvilli in all your cells? Do all your cells have microvilli? No, because some of your cells have really nothing to do with rapid absorption. But cells that need them, need them. And if they don't have them, this is going to be a huge problem. Let me give you an example. Does anybody know where in the whole 30-foot tract of the GI system, do you know where most of the absorption of your nutrients occurs? In what section? So do y'all want to hear? Y'all want to see me draw? Because I know y'all like. So you've got a mouth, right? You've got an esophagus going to a stomach. Is that right? Is that going to stomach? Y'all see that? Yes. All right. So is it in the mouth? Do you think? No. No. Is it in this muscular tube called the esophagus? No. The stomach. This is churn. This churn that food stays in for a pretty good length of time. Is it that? No. No, okay, well, give me a break here a second. And then you've got these small intestines that are about 22 feet. And then you've got large intestines that are about 5 feet or so. Rectum, anus. Okay. So just looking at the anatomy, what would make the most sense where most absorption is occurring? Does it? Don't you love it when it becomes easy? Because why do you say that? Why do you say it makes the most sense that most absorption needs to occur in the small intestines? Why do you say that?
Because it has the most surface.
It has the most, because the surface area was, and it's the longest, and food's going to be in there the longest, isn't it? Going through that. And it's really convoluted. Y'all know the small intestines are really convoluted, 22 plus B, depending. And they're convoluted because the food, as it's going through, because of the convolutions, is going to swirl. It's going to be swirling. And when it's swirling, what's it hitting up against? The microvilli. And what is, you know, because the cells that are lining the small intestines, I could straighten out, who's Queen Chitlin? I could straighten out, me too, Chris. I could straighten out the intestines and look through it, and there's just a, you know, it's just a tube, isn't it? But the inner lining cells have these microvilli, and as the nutrients are hitting that surface area, is that helping with the chance of what? Absorption. Okay, so what's the function of microvilli?
Absorbing rapidly.
Rapid absorption, that's what it's called. So, here's an actual cell. Here's an actual cell where the cytoplasm has caused these protrusions. What's the sugar coating? I like this picture better. This is your sugar coating, the glycocalyx. What's found here? The recognition proteins, right? The recognition proteins are here. The cell identity markers are here. This is your sugar coating. This is on all cells. But this microvilli are only going to be on cells where we need what? Rapid absorption. Don't let me write on this whiteboard with those markers, okay, guys? Don't let me do that. I'll go to do that sometime. Now, look, guys, cilia. Let's now write down cilia, and let's go back to this false cell. Now, these were what? Microvilli. But these projections here, these cilia projections, cilia, let me tell you, they're called hair-like projections. If you call them hair, I'm counting it wrong. Okay, now I'm going to count it wrong. I'll tell you right now. But some cells have cilia on their top surface, and the reason they do, and when you look at a living cell, y'all could actually Google this. You know, back in the day when I was in school, I didn't have that ability. Y'all have all kinds of animations you can look at. It's amazing. But Google cilia living cells, and y'all will see some pictures of cells working. And what they're going to look like to you, it's going to look like, do you remember, you've seen on Discovery Channels and stuff, like CNN and these, have you seen those? Where it looks like, you know, that's what it looks like. And they're moving like that. And it looks like they're actually moving because of the waves and the ocean, right? Emotion. Okay, so they're moving because of that. But cilia, let me tell you what cilia do. If you look at a human ciliated cell long enough, you'll see there's rhyme and reason how it's moving. And cilia are actually moving in a very coordinated fashion. They've got these proteins that are going to be motor proteins that are moving in a very special kind of way to move substances past the cell. Can most of our human cells move on their own? Human cells aren't moving. There's only one human cell that can move, and that's a what? It's only one human cell that can move. Well, red blood cells are being pushed around through a liquid because of a pump, right? But there's only one human cell that can actually move and propel itself. It's a sperm. All of your other cells, your kidney cells, your liver cells, your central nervous system cells, your cells are where they are. And they have to be able to get the nutrients nutrients have to be able to get there and to move away. But sometimes cells need to be moving substances past the cell. So cilia are doing that. Let me give you an example of some areas that had ciliated cells and see if this doesn't make sense to you. See if it doesn't make sense. In your lower respiratory tract, the air we're breathing, is it sterile? No. Y'all see the little things in the light, when you see light coming through windows and you see lights, you'll see these little things. And, you know, those are dust mite feces and skin cells of your neighbors sitting next to you. And those are just things in the air. That's okay. It's okay. There's pollutants in the air. There shouldn't be too many pollutants. But are we having to breathe sterile air? No, of course not. And so we can catch some of the things with our nose hairs. Nose hairs are a good thing. up to a point. Okay, so in the nose here, and our nose is wet and whatever, and so some things get stuck in the wet and gooey. Then you swallow it down your esophagus, so you're swallowing your neighbor's cells and your that's my easy. And so most of that's happening. But then some things actually get to the lower respiratory. Is that right? Lower respiratory. But we have in our bronchi, y'all all heard of bronchi, haven't you? We have in our trachea and bronchi, we have these ciliated cells that are through this nice saline mucousy layer, are trapping things and moving them like an up escalator. And most of us, we don't even know we're doing this because we don't actually have to do. We don't even have to do that because it's actually moving over the glottis, top part of the trachea, and going down your esophagus and just getting cleared out. So that up escalator, it's beautiful, right? So cilia act as projections that move substances past the cell in a very coordinated fashion. Ladies, you have 2 ovaries, right? And each month, 1 ovary would get targeted for ovulation if you're not doing any sort of birth control, whatever. And so the ovulation will occur, and that little egg has to be fertilized within a couple of hours or it dies. The sperm had to move all the way up through the uterus, through the fallopian tube, out there to the ovary, right at the edge of the fallopian tube. So now you got this zygote that is now moving down through the fallopian tube to get to the uterus. It takes that newly developing group of cells 6 days to get down there. Do you think gravity takes it down there? Or do you think inside the lining of your fallopian tubes, what do you have? Cilia that are moving it there, right? So cilia function by moving substances past a cell. If you tell me that it moves a cell in humans, I'm going to tell you that you're wrong and you're going to miss those points, right? So in cystic fibrosis, what ends up happening is these ciliated cells, instead of having a nice saline layer that can move things quickly as an up escalator, it gets what? Is mucus a good thing or a bad thing? It's a good thing. It's good as long as its consistency is what it needs to be. If it's too mucousy, is that a problem? Yes, or if there's too much production of it, there's a problem. So this is what happens in cystic fibrosis. And I told you it's because of faulty what? Do you think you need to know that? Yes. And I also told you that it's a what type of disease? Hereditary, an inherited disorder. So if you were to tell me it's autoimmune, I would tell you what? You're wrong. So don't. Is it important to know causes of diseases? Because and what disease is actually what's occurring. It is really important because if you don't, you wouldn't understand how to treat it, would you? Okay. So great. A flagella. Flagella is going to be the only human cell that has this is a sperm. And the sperm does have to move, right? And so it has a long way to go, and it really does, sperm do. And so we'll talk more about that in the reproductive system. Okay, so now what I'd like to do, and then I'm going to give you a break, I want you to write down in your notes, I want you to write down 2 words, diffusion, and the other word I want you to write down is osmosis. And go ahead and write versus osmosis, okay? Diffusion versus osmosis. And here's what I want you to know, even though sometimes even in literature you'll see this differently. You'll see this as a confusing thing. But here's what I want you to know. Diffusion is going to be referring to the movement of solutes, where osmosis is going to be referring to the movement of water between 2 areas of concentration. So diffusion is solutes moving between 2 areas of concentration, and osmosis is movement of what? Water. And you might be thinking to yourself, solutes, like what's an example of solutes? Here's an example of solutes. Anything that's not water. that's a solute. Oxygen, O2. It's hard to think about O2 as being something real, isn't it? It's a gas, it's tiny. And how can that be real? It's a solid. CO2 is a solid. Sodium, solid. Potassium, solid. Glucose, exactly. H2O, not a solid. It's water, right? So, okay, let's take a 10-minute break. We'll come back. Get up and jump around or do something. Okay, are most people back? I think, okay. So guys, diffusion versus osmosis. So we are thinking about, and I often use this room as like an illustration of one human cell, right? So we are the intracellular substances inside the cell, and like anything in the hallway or outside this window or whatever is extracellular. Fair with me? Is that fair? All right. So we know we're filled with fluid. We know we've got all kinds of solutes, not just water, but solutes inside of the cell. And we know that there's a lot of them outside, and that water is bathing the cells, and there's a lot of solutes outside. So sometimes things need to come in, and sometimes things need to go out. This is what we've been talking about. So what I want you to know is that when we think about solutes, even small little gases, and how they move, and electrolytes, which y'all know, sodium, potassium, chloride, y'all heard of some of these electrolytes, how they move in and out of the cell, these solutes, is by diffusion. And what I want you to know about diffusion is that things move, these solids move down their concentration gradient. Now what that means, that's a fancy way of saying that if there's a lot of them outside, but not many of them inside, they would want to do what? They would move from high concentrated areas to lower until there was this balance between the two areas. The membrane is keeping the distinction between the two concentrated areas. Is that good for you to understand? So the way solids would want to move is down their concentration gradient, and they would be trying to do that. Just like if I took Megan and I had a bottle of perfume that has a lot of concentrated perfume in the bottle, right? And I spray it, Megan, can you smell it? You can smell it right now. But can you smell it, Kendra? Not yet, but in a few minutes, you might be able to start smelling it. What happened to that solute? The solutes that were the perfume, what happened to them? Megan could smell it immediately, but it took you a little bit to be able to smell it. What happened to those solutes? They moved from high concentrated areas to lower. Do you get what I mean? Did it take any energy? Did I have to stand up here and do like this for that to happen? No, they're just gonna diffuse from high concentrated areas to lower. Like if you put a little crystal dye into an agar dish, all the agar looked like this at first. All of it looked like this. And you put a little crystal in that was really dark purple. What started to happen in that agar dish? The dye started to move out, right? From high concentrated areas to lower, just like perfume moved out. Hey, Kendra, put it back in the bottle. Bring it on back up here. Any of y'all smelling that perfume? Who's smelling the perfume? Play this game. Thank you. Thank you for playing. Y'all bring it back up here and put it in the bottle. Bring it from the low concentrated areas. Bring those solutes, because those are solids. Those are molecules. Put them back in the bottle. So you can't move solids up their concentration gradient from low to high. You can't do that? You can. Living cells can. Living cells can move solutes from low to high. But is it going to take work? Yeah. Are y'all going to have to devise some sort of vacuum? Are you going to have to devise some sort of something that would separate it out from all the other stuff? And are you going to have to purify it? It would take work, work, work. Moving down the gradients, did it take any work? No. No, that's just passive diffusion. It's called passive diffusion. They're going to want to move that way. But as a cell, think of us as a cell, guys, now. Can we just let any solids move in willy-nilly? Just because it's moving down their gradient, or we're going to have a way to keep them out? Are we? Are we going to have a way to move solids against their gradients? We are going to have. Is it going to take work?
Yes.
That's called active transport. And the way the cells do that are with pumps, and are with carrier molecules that we are constantly doing that. That's going to take work. What molecule is that going to take? ATP, a charged battery. You start running out of ATP. I told you all, you have about 4 minutes, don't you? And cells start losing, with all the work that's being required, they start losing the ability to move the solids to the concentrations they need. And do you think that's going to affect the cells? Yes, cells are going to start to die. How do solids move naturally? Down their concentration gradient. What is that called? Passive or simple diffusion. Passive or simple diffusion. But can living organisms, can cells do work?
Yes.
Which one of the many ways they have to work? By moving what? Solids up their gradient. What's that called? Active transport. Are y'all good? All right. Now, the reason that it's so important that you understand the distinction for how solids move versus how water moves. Water moves exactly the same way. It moves down its gradient. If there's a whole bunch of water outside our cells versus what's inside our cells, if the water balance isn't what it needs, isn't equal, water will move into the cell. You know what the difference, though, in the movement of water versus the movement of solids? Your cells can't do a damn thing about it. And I say it like that because I want you to wake up, and I'm sorry if that offends anybody. But I'm telling you, they can't. So if there's a ton of water outside the cells and not as much compared inside the cell, where will water move? And will the cell be able to do a thing about it? So the cell's going to do what? And the cell's going to change shape. And it might expand so much that it does what? Burst. Water moves down its concentration gradient, period, until it's equal between two areas of concentration. And there's nothing that living things can do about it. The movement of water is called what? Osmosis. So you see that that's a very different way than living cells have the ability to move solids up their gradient, don't they? Don't they? With energy, has the ability to do that. And I just can't stress that to you enough, that that's a big difference. Now, here's what I hate about your textbooks. Oh, I think I took it out. Maybe I took it out. Oh, so I think I did, because in your textbook, it'll say osmosis is the diffusion of water. And I'm thinking, don't even put that word with water. It's the movement, it's the flow of water. So guys, let's look at this. Let's say that this is inside, let's say that this is inside of our cell. This is a cell. And let's say that this is the fluid bathing our cell. You with me? So side A is what's inside of a cell. Side B is the outside water, the outside fluid that's bathing the cell. You fair enough? These little dots, big dots, little dots, represent solids. Which side has the most solids? And if they have the most solids, but we look at this water thing, that means it has less what? Because these solids are displacing where water would be. So there's more water inside B. Would y'all agree with that? So what is going to happen? What is going to happen? What's going to happen? It's going to be osmosis. Side B, the water's going to move into side A until it can't move anymore. It might be that it can't move anymore because of this hydrostatic pressure, but it's going to move until it's a balance between the two areas. So that might not be good. Is that right? That really might not be good. I want you all to write down the term osmolarity. Have you heard of the term osmolarity before? How many of you have heard of it? It should be everybody, because everybody in this room should have had chemistry. So everybody in this room's had chemistry. What's your definition for osmolarity? That many definitions. You've had chemistry. What's your definition for osmolarity? When you're talking about milliosmoles or whatever, and you're talking about osmolarity of something, what are you talking about? Every one of you had chemistry, but not many of you had definitions for pH, which is a little concern. But anyway, what's osmolarity mean? Come on, I know somebody knows it. They're just sitting back there being quiet. No?
Is it reverse osmosis?
Osmolarity means, and just listen to me first, then I'm gonna let you write it, okay? When you're talking about osmolarity, it means that you know how many solutes are in that solution. It means that you know the percentage of solutes in that solution. Hey, by the way, if something has a high osmolarity, it means it has a lot of what? Solids. If it has a low osmolarity, it means it has a lot of what? Water. Did you get that? Osmolarity means that you understand the percentage of solutes in that solution. And if you understand the percentage of solutes, by God, you understand the percentage of what? Water in there. Are you good with that? So that's your working definition for that. Don't memorize it like you did in things in chemistry and then take them out of the classroom. Learn a working definitions, okay? So osmolarity. So let me say this. Do our cells, inside of our cells, have a certain osmolarity associated with them?
Yes.
Does the fluid that's bathing our cells have a certain osmolarity associated with them, with that fluid? Does our plasma from our blood, which is the liquid part of our blood, have a particular osmolarity that should be present? It does, because if they aren't all balanced, what's going to be moving? Water. And it's how water moves between these components. When I, in my 30-foot track, drink water, like, where's your shot of water bottle? I got coffee. Y'all might think it's something else. It's not. It's coffee. It's black coffee. But if I drink this water, are there no solids in this? Okay, look. I was looking to see if somebody had something I could see through, like a Mountain Dew or something. Just hold that up. Hold that up. Y'all probably don't like me touching your phone. Okay, which one do you think has the most solids, has the highest osmolarity? Which one do you think? Can you always tell from the color? No, you can't always tell from the color. But in this case, y'all are looking at this and saying purified water. So it's got a reduced number of solids, but it's still got a lot of solids, but that's got more. It really does, right? So that's got more. So here's the thing. Here's the thing I want you to be thinking about. If water is different from our blood plasma, the osmolarity from our blood plasma, which is sending nutrients to our tissues, because our tissues are stable, the cells can't move, can they? Then it means that if the osmolarity is not balanced between, water is going to move into the tissue spaces. And then that osmolarity is going to affect the intracellular fluid, and that's going to be affected. Would you agree with that? Does anybody know what it's called when you have too much fluid in the interstitial spaces between your cells and your tissues? Does anybody know what it's called when you have too much? Edema. Edema. That's exactly what it's called. Edema. When I drink water into this water, which has very low solids, low osmolarity, I drink it into my 30-foot track, it's a lot of that water is going to move into my bloodstream. And from the bloodstream, is it just going to stay there? Or is from the bloodstream, it's going to also move into my tissue spaces? And from the tissue spaces into my cells? And is water going to be flowing sort of back and forward? Is it? And should we have osmolarities between those areas balanced? Because if we don't, water might aggregate, if you will, in certain tissue spaces. If you have edema in your toes, ankles, and your fingers, it might be unattractive, but it's not killing you. But where can that edema end up going? To your lungs. And if your lungs have too much fluid in them, can they change shape? If they can't change shape and size, they can't move what? Air, right? If there's fluid around your heart can't change shape, meaning it can't fill with blood effectively and pump effectively. Your heart and your lungs are in these compartments that they have to be able to change shape and structure. because they're filling with air and releasing air, and the heart's filling with blood and pumping it. If they've got fluid around them, they can't change shape. So you've got this sac that the heart's needing to fill and pump, just like lungs are needing to do, but all of a sudden the sac is doing what? Squeeze them. And now can the heart do it? It can't do it. Do you get what I mean? So edema, you can have tissue edema, but you can have pleural edema. What would plural mean? Lungs. It's around the lungs. If you have pericardial, peri means surrounding, right? Cardium is the heart. Pericardial fluid. Does that sound like that would be a good thing to have an excess of that? No. So edema. You can have cerebral edema. Bless your heart. You're on your way out. Right? Because cerebral edema means that there's now fluid surrounding the brain. The central nervous system is shutting down. Okay. Do we care how water moves? Yes. What is the movement of water called? What is the only way that water can move? Down its concentration gradient. Do you have a working definition for osmolarity? Yes. I want you to write down the word tonicity. I want you to write down the word tonicity. Write down tonicity. Tonicity is referring to the ability of a solution to affect the volume size of a cell. Is that what I've just been talking about? Is this just what I've been talking about? So tonicity is this idea that some solutions, depending on their osmolarity, can affect cell size, So here's what I want you to know. You have this in text format, but I'm going to show you a visual because I told you I like visuals. All right, look, guys, prefixes. What does hypo mean? Low. What does hyper mean? High. What does iso mean as a prefix always? Equal. Is that right? I want you all to look at this normal red blood cell. This is, again, a scan of a normal red blood cell, an actual human red blood cell. And this is the way it's supposed to look. Red blood cells are said to be disc-shaped or sometimes referred to as donut-shaped. Can you see why they're called donut-shaped? Do you think red blood cells really have a hole in the middle? They do not. They have their, if you were looking at this from the side, it's just that they get thin in the middle. Do you get that? So when you're looking at a scan of it, you sort of see a shadow there where it got what? Thin. They're disc-shaped. All right? This is a normal red blood cell. And in this normal red blood cell, you put normal saline, which is isotonic. Isotonic. Meaning that the fluid on the outside is going to equal the water. It's going to equal to the inside. So is water not moving between these two, between this membrane? It's moving. Water's moving. Water's moving in, and at the same rate, it's moving what? It's just moving, but it's not affecting the cell size. For a red blood cell to work, it better look like that. That's how it needs to look to work. But what if we put this very same red blood cell, we dropped it into a hypotonic solution? A hypotonic solution has a low number of solutes, which you told me means it has a high amount of what? So a low number of solutes means there's a lot of water, and how is water going to move? And what's that cell? Is it going to change the cell volume and size? Is it going to change how the hemoglobin inside that cell can function? I guarantee you it is. That red blood cell is not functioning. It is not. It even is at risk of doing what? Bursting. It is. All right, now, suppose I took this very normal red blood cell that's doing what it needs to do, and I put it in a hypertonic solution. In a hypertonic solution, there's a lot of solutes. That means there's a low amount of what? Water. So now there's more water inside the red cell than there is outside the red cell, and water, where will water move? It will move out. And now this cell did what? It shriveled. It shrunk. It crenated. It crenated. And that's what crenated means. It means it shriveled, it shrunk, And is this red cell functioning?
No.
No. So you've essentially, if you change with tonicity the fluids that the person has going in, can you actually be changing their cell volume? You can. And is that going to be changing their functions? And are some of them going to be losing their functions? Yes. Okay. So guys, here's the goal. The goal is to stay well hydrated. The goal is to not put fluids in a patient that you don't know what you're doing, because can you compromise them? Yes. Can you compromise their lung capacity? Can you compromise their cardiovascular system? Can you overwhelm it? You can. One of the last patients that I worked on clinically was a patient who had a GI bleed. This gentleman came into the ER and it looked like a river of blood. How many of you have seen GI bleeds that like happening like that? They are losing. You know, your GI tract is sort of like a mouth injury. A mouth injury can look horrific, but why is that? Well, they can be horrific, but what you say?
You don't know where it's coming from.
You might not know where it's coming from, but also there's a lot of fluid in it, so the blood is mixing with that. So you're thinking, is this all blood? You know, it's just like, because that looks like awful. So anyway, your GI tract, you have about 9 liters of fluid in your GI tract at any given time. That's a lot of fluid, isn't it? And then you have a GI bleed that's mixing blood with it, and sometimes it can really look like they're losing a lot of blood. And they may not be losing as much as you'd think, because it could just really be a lot of mixed with fluid. And sometimes they are losing that much blood, and they can bleed out. It's the real risk of having GI bleeds is bleeding out, and it can happen slowly. And it, you know, so some people can have a chronic GI bleed that takes them out of the world, and some people can have an acute GI bleed, like this gentleman did. And he was losing so much blood that we had given away all of his. I don't take it back because we hadn't had time to do the type across. So we were given O negative. That's packed cells. But anyway, and just signing them out even before even any types and crosses were done. But there was a real concern on my part that this was going to be volume overload, because they were not really, you know, it was just really hard to keep up with where all that fluid was and where it was going and what was really being lost. And so then the risk becomes, and this has nothing to do with the medical community, it's just the nature of the friggin' beast. But there becomes this thing that you don't know if they can handle the fluid that you're putting in. And he ended up dying the next day in ICU from fluid overload. So nature of the beast, it really is. I found out the next night I was in that had happened. But it, you know, he was just really bleeding out. It was like a river leaving, a river of blood in the ER. So, all right. It's important to know what you're doing with those fluids. It's important to understand osmolarity and how things are moving in and out. What I would like you all to know is that when you think about the cell, and that there's a lot of solutes out there with the water that's bathing the cell, there's a lot of solutes in. I do want you to understand that the predominant cation, positive ion, outside of our cells should be sodium, Na. And the predominant cation inside of our cells should be potassium. Now, to keep that, because aren't they wanting to move down their gradients? Aren't they wanting to? Aren't they, guys? Can y'all tell me that solutes will move down their gradients naturally? They are trying to do that, but what's keeping them from doing that is something called the sodium-potassium pumps in all of your cells. This is just another thing that's happening 24/7 in every one of your living cells. You have these sodium-potassium pumps that are actually functioning. Look at this. Half of your daily calories go to your sodium-potassium pumps. If you are not pumping that sodium against its gradient, you don't get to live. Another true story. A phlebotomist brought some blood to me. I was working night shift, whatever. And I told him, I said, look, this is not compatible with life. You drew this above an IV line or something. He was like, no, I really didn't. I didn't turn on the light, but I'm sure there's no IV line. I've been drawing his blood every night. I was like, well, you need to go back. Because he's either dead or you did. Yes. He was dead. And he just had, you know, at night when you're drawing blood on patients, sometimes you don't want to disturb them. So you might not even wake them up. How many of you have done that? Has anybody in here done it where you don't even wake them up to do the blood glucose? You just go and think you can get by with it and they won't even wake up, you know, draw the blood, do whatever. And you can sometimes, and so it's good. So you know which ones you can do without disturbing them, because really we disturb them whole lives. So anyway, but he came over and said, oh my God, he really was dead. But there's some things that aren't compatible with life, and as soon as the cells die, these chemistries change because the pumps aren't working anymore. Are you good with that? But we have these pumps that are usually working, and they're usually doing what they need to do. So, yes.
How were you able to tell that he's dead? Like, what was
His chemistries, his sodium, potassiums were just completely off. They weren't compatible with life. Now, if he'd drawn above a line, there's sodium, there's saline lines, right? So if you do that, they can throw those results off depending on what kind of draw you had. So I had said that to him, but I was really thinking he really had drawn above a line, right? Because I was thinking he probably would notice the person was dead. But I was wrong. And so, you know, whatever. Hey, no, I'm not laughing at the person's not. That's all. So, but no, you get what I mean. Like this wasn't an unexpected death, by the way. I'm not telling you, you know, this person had been there a long time. They were really, it wasn't unexpected. But nobody had noticed it yet. None of the floor nurses had noticed it yet. He didn't notice it because this is the state this person was typically in. You know what I mean? But anyway, the blood work knew, you know, the blood work told us the story. It wasn't compatible with life. Okay, so okay, so we're thinking about the cell. We're still just talking about cell membranes. So we're thinking about this cell, if you will. So this is a cell and something's on the outside. Cells are going to have the ability to take particles and actually bring them into the cell. This process is a process of a cell eating. It's called phagocytosis. And have y'all heard of that before? Phagocytosis, so phage is to eat, a process of a cell eating. If I break down that big word, remember there's no such thing as big words, are there? They're just prefixes, suffixes, and root words that you just interchange and whatever. So the process of a cell eating. And so they bring this in, and then the cell's going to have the ability through lysosomes, which I haven't told y'all yet exactly, lysosome vesicles, but I will. Lysosomes, actually go ahead and write it down now. Lysosomes has digestive enzymes. Inside of all of our cells, we have these lysosomes that contain digestive enzymes. And one of the functions of them is to actually, through bringing particles in, these lysosomes can hook up with them, tear down that particle, digest that particle, use anything that it needs and wants, and spit out the rest. These lysosomes work on a lot of different levels, these vesicles that our cells have that have these digestive enzymes. Sometimes these lysosomes are working on your own organelles inside of the cell by recycling them, destroying them, breaking them up to monomer units, and then you recycle those monomers and make new organelles, right? Sometimes these lysosomes will hook up with foreign particles that are deadly and destroy them for you. So they're protecting you, aren't they? Lysosomes also play a role in cell death. When the cell has lived, and the DNA determines it, when the cell needs to die, I told you some of your cells will only live for hours, and that's all they need to live. And when they need to die, do they need to die? Yes. Do they need to get out of the way?
Yes.
Yes. Are y'all good, Kelly, Alexis? You good, Alexis? So when cells need to die, they're going to end up making more lysosomes are going to be created, and it's going to really eat itself. It's going to auto-lyse. Auto means self, lyse means to split. It's going to self-destruct. What happens to cells that lose their mechanism, their life cycle mechanism, and don't know when to die, and don't know when to stop growing, and don't know which shapes to take, and just steal everything from the environment because they lose that? What is that called? It's called cancer. Those are cancer cells, aren't they? That have lost their guidelines, if you will, for when they are supposed to. It's important, even within an organism, it's important that death is a very important part of the life of the organism. Inside of you, you have cells dying all the time, and they better do it when they need to do it. They need to get out of the way. They need to be replaced, and they need to be working optimally, right, to do that. So death is an important part of any ecosystem for life to be able to continue to exist. It's even important inside of a living organism. You see what I'm saying? Okay. And lysosomes play a role in that. Now, that was a little illustration, but here's an actual electron micrograph. So this is a cell membrane. These are particles in the extracellular fluid. They hook up with receptors that are in the membrane, and then the entire membrane collapses on itself. And now, what had been part of the external membrane is now vesicle inside the cell, and the cell can break this open and use those particles it brought in. So this is a process of endocytosis, the process of cells bringing things in. And the cell has the ability to do the opposite of that, where it makes something inside the cell, sends that vesicle up, launches it to the surface of the cell, then releases it. So this is exocytosis. Fair enough? So endocytosis versus exocytosis. Good question, Rihanna? Endocytosis is this process of a cell bringing things into the cell. Endocytosis, a process of a cell receptors having things attached, and then the cell membrane collapsing, so that now what was outside the cell is now where? Inside the cell. But the reverse happens as well, where cells make products. I told you your cells are factories. This launches up to the membrane surface and then excretes. This is an actual picture of a vesicle that had been made inside the cell, going to the surface of the cell and now releasing its products, right? So our cells are all secretors. They really are. They can all secrete. And now we get to go into the inside of a cell. I'm going to go for just a little while more with this lecture. I know you guys probably feel like you have plenty of notes. But this next part kind of goes a little bit faster, if you will. But I had told you about the cytoskeleton. What are some words, descriptors you had for these little webbings here? What words do you have? Okay. Okay. But what functioning kind of, they look a little bit like that. What'd you say? It's like the highway system, so moving things inside the cell, so moving products to the surfaces or excretion, right? So movement of products all throughout the cell, scaffolding, so form, structure, that kind of thing. They really work for that. So that's the cytoskeleton. Here's the picture I wanted you to see. Again, a scan of a cell. This is the nucleus of the cell. Do y'all see the cell? This is the nucleus of the cell. But look at this. Do you see what I mean? It's like kind of cool, isn't it? Okay. Now organelles. Here's the thing about organelles. Really? Okay, wait. Well, that looked right there, but it's actually here. Okay. So a nucleus, guys, a nucleus of a cell. So if these are highlighted little nucleuses so that you can see, you know, what it looks like. I actually want to do this a different way. And I'm just going, because this next part moves kind of faster, if you will. And I want you to bear with my drawings a second. So we said plasma membrane. We know the functions of plasma membranes. We know what they're made of. We know what molecule helps to carry out their functions, mostly proteins, right? We know they're made of mostly lipids, but the proteins go from. We know some of them have these things that move things past the cell. They're cilia. We know some of them are going to have these extensions that help with absorption. What were they? Microvilli. Is that right? We know they all got a sugar coating. What was that? Glycocalyx. We know there's one cell that has an external motor protein called a flagellum, right? And what was that? All right, so great. Now, cells. Cells, our complex cells, sometime in their life cycle, are going to have an area that has a very specific function of housing the nuclear DNA. This is the nucleus of a cell. The nucleus of a cell has the function of housing the cellular DNA, organizing it, and this is where it's going to be. This is like in a business, this would be the administrative office, right? So you can think that. And by the way, do I talk about cells as being factories? Because honestly, they are. And factories are businesses. And factories don't make anything until they've been given the what? Orders. Is that right? Would y'all be good with that? So what would you tell me in your own words a nucleus function is? To do what? What'd you say? The nucleus function is to what? House the DNA. This is the administrative office, right? So this is housing the cellular DNA. It's where the whole big cookbook is. Now, when I drew this circle around this, there's an actual membrane keeping this in and things out. And yeah, there's still some diffusion and all that stuff happening. Of course there are. But this is called the nuclear envelope. Because what is it holding? It's the DNA inside the nucleus. That's right. Now, this nuclear envelope does have some pores in it. Pores allow for what? Things to move in and out, right? And what I want you to know is attached to this nuclear envelope is another organelle. And this organelle is called the Endoplasmic Reticulum. This is called the ER, which you all would want to know is the Endoplasmic Reticulum. So this Endoplasmic Reticulum has a rough part and it has a smooth part, smooth ER. The rough part is called rough because even on electron micrographs, it looks what? Rough. And this part looks smooth. Let me tell you why this looks rough. These little dots that make it look rough, these are called ribosomes. You with me? Everybody with me so far? Are y'all good so far? All right. Where did you say for everything that's happening inside the cell, depending on whether it's going to have cilia or microvilli, depending on which size it is, depending on how long it's going to live, depending on what it's going to make, where did you say all that information is coming from? But the DNA molecule, hold it up. The DNA molecule is huge. It can't get through this nuclear envelope, but what can get through it? What smaller molecule that holds only one? Oh, look at one little recipe. One little recipe. What one little recipe can get through? What is this molecule? It's RNA. RNA can get through. And guess where RNA can go when it gets through? It can go to the what? And guess what these are? These are workbenches. Where what comes to them? Workbenches. What comes to this workbench are the little RNA. The little RNA, messenger RNA. It comes to this workbench. One recipe at a time comes to this workbench. These ribosomes that are on what membrane bound organelle? On what one? Endoplasmic reticulum. And it's got so many ribosomes that even on an electron micrograph, it looks what? Rough. Because there's so many workbenches, so many ribosomes. What's coming here from the DNA? A tiny little recipe coming to the workbench. What's that going to be a recipe for?
Everything.
I know everything, but what did I tell you your cells are essentially? So this RNA comes here holding a recipe. Is that right? And you said there what? Tell me again. For proteins. So this is going to be where proteins are getting what? Made. Happy. Because proteins, we've already seen how many functions they have in the plasma membrane, haven't we? We already know that inside the cell, there are going to be proteins that need to act as catalysts. What is that? Speeding up reactions. Speeding up all these reactions. Everything I'm about to tell you happens really quickly. Because of what? Catalysts. A category of proteins called catalysts. So these are where proteins are being made. Ribosomes are where? Or what? Or what, I mean? Workbenches, where what's being made?
Protein.
And where did the recipe come from? Which came from where? Are you good? All right. Why does this look smooth, and what function could that have? Let me tell you something. Have you ever used lard? Does any of y'all ever use lard? Or Chris says he's cleaned chitlins. That means he's cut up as something. You've done your own meat. You kill pigs. You've done your own meat. So you know your hands start to look what? Animal fat looks slick, doesn't it? It looks slick, y'all, and it really does. And animal fat, this is where the lipids are made. Don't we need lipids inside of our cells? Don't we need lipids? Aren't lipids fats important? Yes. But y'all all know lipids look slick, don't they? And guess what? They're looking slick here too. And that's why this ER looks what? Smooth. So if I were to ask you the function of the smooth ER, you'd say what? It's making lipids. If I were to ask you the function of the rough ER, you'd say what? Building proteins. If I were to ask you the function of ribosomes, you'd say it's the what? Workbench. Where what comes? The messenger RNA comes. So that proteins can be built. Are you good? What is what?
The rough ER.
The rough ER has these ribosomes on them, the workbenches where proteins are built. Proteins are built. All right? So y'all are telling me that directions from the DNA are being sent out to the factory. Proteins and lipids are being built. Is that what you're saying, Kelly? That's what you're saying. Are they all just staying right there? Because they're building up now. Do you see them? Do you see the proteins? Proteins building up. Back where there's a smooth, lipids are building up. They just stay right here? No. They actually are going to go to an area. Okay, my drawings might leave a little to be desired. But these products are going to, these proteins, these lipids are going to go to this Golgi apparatus, Golgi body, Golgi complex. I don't care. As long as you know it's Golgi or Golgi, it's okay to say too. This Golgi, let me tell you what it's going to do with these products. It's going to wrap them in a vesicle and ship them. Because you all know that these don't just jump hop here. They're being moved by through what kind of highways? Cytoskeletons. And there's catalysts moving them to where to be wrapped and shipped. Where are they going to get wrapped and shipped? The Golgi, right?
And that's the proteins and the lipids.
Yes. Any products being made have to go there to be wrapped and shipped. Now, some of these products are just going to be used in the cell. They're going to be just sent straight to be used in the cell. But some of them are going to have to be wrapped and shipped. And then do you notice these vesicles, they might have to be sent through exocytosis and leave the cell. Do you get what I'm saying?
Yes.
Right? This is sounding like work. Is it sounding like work? Is it sounding like work? And that's okay that it's sounding like work. because we have throughout, we have these organelles that are called the what of the cells? They're called the powerhouses of the cell, because what is being produced here? These are mitochondria, and this is where ATP is being produced in all of these mitochondria, ATP, because it's all of this taking work. You said a lot of the ATP is being used for pumps and for channel proteins and for whatever, but are we needing ATP to do all of this work too?
Yes.
Are we? Are we?
Yes.
Now we want you to know there's also free ribosomes all throughout the thing, and they're just ribosomes, whatever, which are what? Workbenches where proteins are being put together. But the endoplasmic reticulum is where most of these are being made, and the smooth ER is where what's being made? Lipids. And then they're being sent to the where? And the Golgi is doing what? Wrapping and shipping. And then that's being wrapped and shipped to different parts of the cell, but some of them to exit the cell, isn't that right? And that is all taking work, which is being provided by the what? Mitochondria. Right? All right. All right. Now, I want you to know, I've already told you that some of the vesicles that you have in your cells, some of them are lysosomes. Didn't I say that? And what did I say lysosomes are going to have inside of them? Digestive enzymes, which are proteins that eat and break down. Lysosomes have a lot of different functions, and I told you some of those functions, didn't I? It's they clear, they clean, they're recycling. They're necessary inside your cells. They're absolutely necessary inside your cells. Have you ever heard of Tay-Sachs syndrome? Tay-Sachs syndrome is a inherited disorder of, it's mostly seen in a group of Jewish descendants of a particular order. But this particular inherited disorder is devastating. The child does not live past 3, 4, 5 years old. And they don't know sometimes until the child is out of toddling, and they're progressing. They're meeting every milestone. There's no outward knowing that this is happening. But what's happening is the lysosomes are not functioning the way that they're supposed to in clearing some of the substances in the central nervous system, so it becomes devastating. And it's a progressive terminal death, early death, and it's supposedly horrific. I've never seen a patient with Tay-Sachs, but y'all have an insight about it. But where is the problem? At what level? Molecular, cellular level. Well, molecular and cellular level is what I'm saying, because our cells are made of what? Molecules, and these organelles that have to function, don't they? They have to function. So anyway, we need them. Now there's other vesicles, and you can't tell from just looking at electron micrograph, but there are other vesicles called peroxisomes. Peroxisomes. You guys, peroxisomes are going to contain antioxidants. Is that a good thing or a bad thing? That's a good thing, isn't it? So peroxisomes, good thing. Now, let me say this. That looked like a crazy drawing. That just looked like crazy. But did I give you the basic functions of these? Did I? All right. I gave you the basic functions of these. And that's fine. I'm trying to figure out what else I want you to know. I want you to know a couple of terms here. I do want you to know a couple of terms. What did you tell me the function of the nucleus is? It houses the cellular DNA. Didn't I say that? Right. Now, I want you to know that when we are looking at cells, we're going to look at some cells next week under the microscope. And some of the cells, when you see the nucleus, it's going to look like, it's going to just look like granular, kind of like to your what you can see with that magnification is going to be kind of grainy looking. That's DNA that is all spread out and in its regular form, ready to function. And DNA, when it's like that, is called chromatin. So I do need you to know that that word, that chromatin, and that's in your notes. I mean, I just saw it. So anyway, chromatin, it's the DNA. It's the DNA that's kind of all spread out. It's in its functioning form. It's called chromatin. But before DNA can actually, before a cell can actually divide and become two cells, what must DNA make a copy of? Itself. So you're saying that before a cell can divide, it's going to have to make an identical copy of this recipe book, the DNA. Is that right? Mm-hmm. And when it does that, it's going to make a copy and it's going to condense. And now when you see the DNA in that form, in copied, condensed forms, that's called a chromosome. And y'all have all heard of chromosomes, haven't you? So DNA and DNA, we said, when it's all spread out, right? It's in chromatin form. It's a double helix. I know that's really a double-stranded, twisted thing, and that's DNA. But before this cell can actually divide, this is going to have to make a copy of itself. It's going to have to make a copy of itself, and this whole thing is going to have to condense down. And so a lot of times you all see DNA that looks like a little X form. You've seen it look like this, and sometimes where it's connected is way up here. And some of them are short, and some of them are little, so whatever. But y'all have seen DNA in this form, right? And this is called chromosomes. These are called chromosomes. How many chromosomes should a human typically have? How many?
23.
43.
No, I'll just tell her. Oh, there's a company called 23andMe.
Because it's not 20. Okay. No, I love that. Okay. And so I can see. Humans should have a total of 46. A total. But wait a minute. This is for the 23. Wait a minute. For your nucleus, for your DNA, for your recipe book that holds all the information for all of your traits, you told me that half of it came from where? So half came from your mom and half came from your dad. Is that right? Mm-hmm. For any, not just us, we like to think it's all about us, but it isn't. For any sexually reproducing organism, it means that two parents contributed to the DNA, 50-50. So here's what I want you all to know. What we do need to realize that what's normal. And when I say what's normal, I'm not saying, I just say what's most have 46 total chromosomes. But we really can't think of it that way, because for every trait, you got a chance at that trait from your mom, and you got a chance at that trait from your parents. So we don't really think of them as total. What we think of them is a paired number, and really, it's 23. It's 23, right? Well, that end is 23. Actually, I wrote that wrong. But anyway, so it's 2 of each of these. So we think about there being 23 pairs. Now, I want to give you more information in case there's any confusion about this. So what it really means is that the chromosomes, so we know when we talk about chromosomes, DNA is in its copy condensed form. Is that right? So what we know is we have 23 of these things in us, right? 23 pairs. This is just the first pair. So this is the first one. Then we have a second pair. Then we have a third pair. And we have 23 of them. So this is the first one right here. We know our 23rd pair is sometimes called the what? What'd you say?
Sex.
Well, not sex genes, but sex chromosomes. And we know that this 23rd pair, you might have on the 23rd pair, you may have a chromosome that looks like this, and you may have another one that kind of looks like this, just macroscopically looks like this. And that would mean you had 2 X's. Is that right? And 2 X's would equal to what kind of gender? Would be equal to a female. Are y'all good with that? All right. But you might have on the, this is or, you might have on this 23rd chromosome, you might have an X, but then you could have this little one here. And this is called a Y, right? An X and a Y. And what is this? This is me. Fair enough? So this 23rd pair are called the sex chromosomes only because, and this is a big only, is because it is on one of these that gender is determined. Gender is determined. But is that the only trait that's determined in the 23rd pair? No, there's tons of traits on these chromosomes. There's traits about color, being able to interpret colors. There's traits that influence your cognitive ability. There's traits that influence your blood clotting factors. There's a ton of traits on the 23rd pair of chromosomes. And you all know that the first through the 22nd pairs, if this is the 22nd pair, and by the way, they're kind of labeled by their sizes. The first pair is really long, and they're joined near the center. The second pair is really long, and they're joined a little bit closer to the end. And they just get shorter and shorter and shorter. But this first through the 22nd pair, these are called autosomes. These were called, what were these called? Sex chromosomes. These are called autosomes because all the rest of the traits, they're found here, and you got one of these from your where? And you got the other one from your where? And how many, for any traits that are found on the first through the 22nd pairs, how many chances at the traits do you have? Okay, let's say there's a trait for hitchhiker's thumb. How many of you can hitch a ride? Some of you can't do that. Some of you are like that. Oh, you're not going to hitch rides. So, is that how you can do it? Really? Okay. Yeah. Who's got a good hitchhiker's thumb? Okay, so you go get a ride, Kendra. So, but all right, don't hitchhike. Okay, no, I didn't say hitchhike. I really didn't mean that. But okay, look, for traits like widow's peak, who has a widow's peak? Okay, so widow's peak, absolutely. For traits like that, they're found on these autosomes, and the trait is found in a very particular, let's say it's on the first pair, and it's in a very particular region of that chromosome, and the trait might be here from your mom, or it might be here from your dad, or it might not be there at all, right? But how many chances at least do you get for a trait? 2, because you would have gotten a chance from your mom's chromosome or a chance from your dad's chromosome. Is that right? Is that right? Okay, that's right. Now, some traits are influenced by several different pairs of chromosomes, and those are the kind of traits that we have for skin tone. Because is there really just a there or not for skin tone, or do you have a rainbow for skin tone? Look around this room. Are there any 2 people with exactly the same skin tone? No. How about hair color? How about eye color? That means that there's lots of chromosomes influencing it, and therefore you get a rainbow of an effect for those traits being expressed. But some traits are very specific, and it's only one location. But do you really just have one location, or did you get 2 chromosomes for that trait? You got one from your what? And one from your, so you get chances, at least 2 chances for those traits. Are you with me? All right. And when you talk about chromatin, that's the cell where the DNA is not in duplicate form, and it's not condensed, ready for the cell to divide. It's all spread out, and it's working. It's ready to work. The cell's ready to work. So what part of the life cycle of a cell do you think a cell spends the most of its time in? DNA, chromatin, or chromosomes? Why? Because the cell's having to work. A DNA is only going to be in chromosome form when the cell is needing to do what? Does that make sense? But y'all have probably only heard of DNA as chromosomes. Have you? Have you? Maybe? Okay, whatever. So I just wanted to introduce that, and that is important information because when we get to the next chapter, you'll have to have a basics of this understanding. Okay? All right. All right. All right, so the nucleolus is actually an area, and let's go ahead and add that to our little list of functions. The nucleolus is an area inside the nucleus where ribosomes are made. Did we say ribosomes are important? Mm-hmm. But what do ribosomes do? Ribosomes do what? They're the workbenches that are going to end up leaving the nucleus, aren't they? The workbenches are going to have to get out of the nucleus. They'll leave through the what? Look at this, guys. Look at this nucleus. Do y'all see the nucleus, nuclear membrane envelope around this nucleus? Do you see the DNA is in chromatin form, isn't it? Isn't it in chromatin form? And here's the nucleolus. But if we looked at the surface of this nuclear membrane, does it look porous? See how many pores there are? And what can move through these pores? What are two things I just told you move through? RNA and what? Ribosomes. I did, and they can do that. And that's what that nuclear envelope is doing. The endoplasmic reticulum, why is the rough? Because the ribosomes are embedded on it, and what's being made there? What's being made there? Proteins are being made there. And what about the smooth? It's lacking ribosomes. It looks smooth to us. It actually looks that way to us on these scans and these graphs. But what did I tell you is being made there? Lipids. That's what you need to know. I'm abbreviating some of it, but not a whole lot of it. So anyway, if we looked at this picture, if we just took the endoplasmic reticulum away from the nucleus, we would see this is the rough and that's the what? Smooth. Now labeling a cell, would it be so hard? Doesn't it look like a factory? Doesn't it seem like that? Okay, so great, and I'm just moving along here with some of these pictures. There are free ribosomes. As I was telling you all, ribosomes aren't just embedded in the rough ER. There are also free ribosomes throughout the cell, and that's true. I do want you to know, what did we say about the Golgi? Go ahead and tell me. What's this Golgi? What's this doing? What are they doing? Wrapping and shipping the products that are made in the cell, throughout the cell, and sometimes even out of the cell. Did we talk about vesicles? Yeah, and we've named a couple of them from the Golgi complex. What did we say? Look at this Golgi. It's all these little vesicles. We said a couple of them are like lysosomes and peroxisomes, didn't we? Did we say that? Yep. And do we know what those functions of them do? Okay. Yeah, we know what they do. So great. Now, we said though that this ends up taking a ton of work, doesn't it? It takes a ton of work. And this is going to be the last thing I'm ending on today. Okay. Going back. And then we'll talk about that. All right. So this is the last thing I'm ending on today, these mitochondria. We have a lot of them throughout the cell. Mitochondria, we know, are called the powerhouses of the cell because this is where the predominant amount of ATP is produced. We can get some ATP just from splitting glucose, can't we? We can get some ATP by sending those products, they're called pyruvate, into the Krebs cycle. We get a little bit more ATP. But where do we get the most of it from is that third step, where oxygen is coming into this mitochondria, being used to create this situation where batteries are being recharged at a phenomenal pace. And those batteries are what molecule? ATP. So ATP, ton of it being produced here. I need you all to know something else about the mitochondria. They are the powerhouses of the cell. They are where most of the ATP is produced. But I need you to know something else. I need you to know that the mitochondria have their own DNA. And it's referred to as mitochondrial DNA. Who's heard of mitochondrial DNA? Where have y'all heard of it? A few of you raised your hands. Where have you heard of mitochondrial DNA? Have you heard of it? There's a couple people raised their hands. You've heard it used, mitochondrial DNA.
This one sounds funny, but didn't they use it on Jurassic Park when the old man was talking about how they reproduce?
Which loves Jurassic Park. Yeah, I love those Jurassic Park shows.
Well, when you talk about DNA, it kind of flashes back.
Yeah, okay. You know, I had somebody in micro, was it yesterday somebody was saying something about The Walking Dead, something brought up in there, they used The Walking Dead. But, hey, guys, fiction comes from what?
Science.
Yeah, a lot of, well, it comes from things that have actually, a lot of things that sometimes have really happened, right? And it uses some of the truth to come up with fiction. My mother-in-law used to have a saying that fiction is never as crazy and as bizarre as real life. You know, people don't even need to make up fiction things because real life is just will blow you away. And she's just so right about that. But anyway, yes, I'm sure you did hear it there. But let me tell you all what it is. It means that mitochondria are not being dictated by what other DNA? Cellular DNA. And it's why I made a point of telling you all that the nucleus houses what kind of DNA? Cellular DNA, which does dictate everything that happens in the cell except for what the mitochondria are doing. The mitochondria have their own DNA. The thing about mitochondria, these organelles, and we know they have their own DNA, if we take them out of a cell, they don't have life. They don't. They don't have life. So they're not allowed unless they're not working and functioning unless they're inside of the cell. But here's the thing. Can our cells work without the mitochondria? So now there is this what relationship? Symbiotic relationship. Is that right? This symbiotic relationship that is occurring. So let me tell you what the idea and the study of mitochondrial DNA. And by the way, there's another organelle that has its own DNA, but humans don't have this organelle. This is the other organelle. Does anybody know what the other organelle and other living plants have it? Other living cells, plants have this. It's called chloroplast. Have y'all heard of chloroplast? Yeah. They have their own DNA as well. But anyway, that we know that there's some organelles that have their own DNA contribute to a scientific theory. Does everybody know what a scientific theory means? It's different than outside of the science class, right? That you think about theory. Scientific theory is called the theory of endosymbiosis. The theory of, and one word, endosymbiosis. And what that theory states, because a scientific theory is really an explanation, isn't it? It's an explanation for a naturally occurring phenomenon. Nature, something to do with nature. And what the theory of endosymbiosis explains is how complex life evolved on this planet from non-complex life. So it explains how 3.8 billion years ago, life, or approximately, they're thinking slower than that now, life started on this planet, but it was single-cell, simple organisms. No organelles inside those organisms. Single-cell, simple life form. How did we get from single-cell, simple life form to cells that have all of these specialized structures that carry out phenomenal functions? How did we get from there to here? But how specifically, see, but how? There has to be, you know, how? And what happened is that these used to be on their own, and other cells used to be on their own. But they started this symbiotic relationship so that with the mitochondria producing a lot of ATP, could more functions occur in that cell. Protists are single-cell organisms too. Protists. But they're way more complicated than bacteria. But they are still single-cell organisms that have mitochondria, that have organelles, that have all of that, but they're single-cell. And then from protists to algae, which is multicellular, to plants. Do you get what I'm saying? Animals. So the theory of this idea supports the theory of endosymbiosis, which is an explanatory statement in science of how complex life evolved from single-celled, simple life forms. So whatever. Okay. Now. Okay, so do our cells have mitochondria? Okay, you all told me your cellular DNA that's in your nucleus that's dictating everything that's happened to the cell from when the cell will divide to when it will die. Right? From how many generations that will take. So as long as when a zygote was formed, a sperm met the egg, and that zygote was formed, that is you, it was dictated when you would die as long as everything else was equal. As long as you didn't starve yourself to death or step out in front of a truck, it's dictated when you'll die. How many times the cells can divide. It's dictated in that moment from cellular DNA. The fountain of youth is inside our DNA. Have we found it yet? Specifically, no. Getting close, though. Getting close. It wasn't in Florida. It wasn't in the Indies. It's actually inside of our DNA. But anyway, back on track. So our cellular DNA came from our mom and our dad, 50-50. That's why we think of DNA as pairs for traits, don't we? Not just a total number, but in pairs. We have to think of them that way. So 23 pairs. All right? Where do you get your mitochondrial DNA? From your mother. So here's the thing. Your siblings that have the same mother, they have identical mitochondrial DNA to you. Identical. So mitochondrial DNA now is actually being used in forensic science. It's being used to identify family members that, sometimes in devastating, earthquakes and whatever, they're not using cellular DNA, they'll be sometimes using mitochondrial DNA just to see relationships, right, between, because it comes from the mother. And there's a great book by scientists, oh my God, and I just lost the title of the book and the scientist. Lord have mercy. Okay, but anyway, it's great. It's Searching for Eve, I think is something like the title, Searching for Eve. Because the fact is, since all of your siblings that came from the same mother have exactly the same mitochondrial DNA, and mitochondrial DNA is evolving at exactly the same rate that cellular DNA does, so we can tell from mutations that occurred how far back they occurred, but we can potentially eventually find the original E. Is that kind of, E, whatever. Not the E you might be thinking of, but E, the first, which is really cool because she's all of our grandmothers, right? So, anyway, mitochondria. How do you feel about that? Any questions? Huh? Did that just blow your mind? Like I said, real things. You don't need fiction, do you? Mitochondrial DNA, and this talks more about it, a little bit more about it. And, there's one thing I didn't give you. Let's give it, because I think it's the last one. It's centrioles. These little centrioles, what would you describe those as? They almost look like little rods, don't they? These little, two little rods. These centrioles, the function I want you to know is they play a role in cell division. And that's all I want you to know for right now. They play a role in cell division. And that is all I want you to know for right now, is that centrioles play a role in cell division. So when the cell needs to divide, they're going to be necessary and they're going to be working. And we're going to see more about that in the next chapter. So in this chapter, did you all sense that other than tonicity and diffusion and osmosis, other than that, did you all sense that I was telling you structures and giving you their functions? Did you sense that? And that's right. And so by Tuesday, should you be able to match any functions with any of those structures? Should you be able to? Great. Now, what I'd like to do is this. I have some people who maybe haven't been doing as well as they should. If people haven't passed, any of the quizzes yet, including the one today, because we went over it, or the lab, I want to meet with you. and I'm going to be in my office, and this time is supposed to be our class time, right? So you can't say you aren't available, because you are. But I want to meet with you because I want to see your notes. And what I'd like to do is just try, and I'm not meeting with you to be I'm trying to help you. So I'm going to try to see your notes, see how they look from today. I want to give you some recommendations. I want to maybe show you where the learning center is for tutoring and whatever, because if we don't start now changing what you might be doing to get this information, it's going to be too late in a week. So we got to start now. Now, as far as lab 2, before we leave, before I turn the recording off, can you tell me if there's any question about anything that you're needing to do on lab 2? Yes, Jada. Thank you. What'd you say? I will, Jada. Yes, I will. Jada, didn't have your textbook good. So what page is that? Or 30. It might be different in different editions, but it looks like this. So everybody, this page. What'd you say? What edition? This is it's the 8th edition. So, but it looks like this, and the others too, I think. So it's this picture. Okay, so this is the page. And Jada, thank you, because I want to start with this, and I want to give y'all some examples, and I want you all to come up with some too. Let's just take a couple of minutes to do that. So I told you that using this picture, I am going to do some filling in the blank types of questions. Julie, can you talk loud enough that they hear it on recording? Can you give me an example of a question I could use that would maybe be fair using that one?
The gluteal is also called the buttock.
Okay, so you could say that the gluteal region is more commonly referred to, and I would have a blank, and you would say butt. Okay, all right, because y'all see that, right? Right. Or you could say, using this word, Megan, how would you say the lower back is more accurately referred to as the what region? Lumbar, right? Or sacral, but you usually call it the lower back, the lumbar region. Jada, can you make up one that I might use? I might actually use it. So y'all give me some. Can you use this picture and come up with something? So you can either give a common name or you can give the real name, and the other would be blank. Give me one. Give me an example. Just it's okay. Okay, that's great. So what would you say? What would you say? Okay, so the cervical region is more commonly referred to as the head. That's exactly right. That's how I'm going to do that. Who wants to give another one? Anybody want to make up one? And by the way, cervical means neck. Jada, I'm glad you picked that one, because I have a lot of students that when I've used that one before, they will say something about the uterus. No. The cervix of the uterus is the neck of the uterus that extends into the vagina. It means the neck of the uterus. Cervical means neck. Yes?
I have a question about that. So the cervical, would that be just the neck in general, or the front?
The neck in general. And thank you. That's a very good question, Kelly. It's a very good question, because I do want you all to realize that, sometimes it is just the whole region, not just like
down here it's at the back of the neck.
Yeah.
Where's that?
Yeah, nucleolus back of neck.
The nuchal, the back of the neck. Yeah, okay, well that can be called that too, but the cervical is really the whole neck. And the nuchal, you really kind of think of that upper neck, don't you? Don't you call that? That's what I call that. So, but anyway, I'm gonna make sure that I would give that kind of thing. So if I said the neck and you gave me either one of those, I would have given you credit. You know what I'm saying? But I'm gonna be very careful in how I do it. So I will either give you the region and you give me the common, or I'll give you the common and you give me the real name, the real terminology, right? So are we good about this page? You know what you got to do from this page? Do you? Okay, so here is what, do I need to get to the visible body? What'd you say?
I just put it in my product.
Oh, thank you. Oh, I appreciate that. instead of me. Let's see. So
And then the next page, we have to know this too. For the
no,
just this
I didn't say that. Just that one page.
Just make sure.
Just that one page. Thank you. All right, look, guys. Has everybody gotten the visible body yet? On the visible body, the first little thing that you click on talks. There's just some anatomical terms, and it's just some common. And it's just trying to encourage you to realize that all words are just prefixes, root words, and suffixes. And it's just giving you a beginning little taste of some, isn't it? Just a little taste. So anything that's on there would be fair for me to ask you the definition of. The second thing is directional terms. So some of you that have just recently looked at it, give some examples of those. I haven't recently looked at it, but I'm going to have to make this quiz. So give me some examples you've seen recently on that. Superior versus inferior. So you'll look throughout this text, if you look at certain organ pictures, they'll say this is a superior view of this, or this is an inferior view. So superior means from above, inferior means from what? What's another thing that they give you on that? What'd you say? Anterior, posterior, ventral, dorsal. I don't know if they give you ventral, dorsal, but anyway, anterior, posterior, whatever they do on there, you all know and be familiar, because I'll say, I'll use it in a sentence with a blank, and you'll have to come up with that, okay, the answer to that. But I won't use anything that's not in that visible body for those two. Is that fair? Okay. So lab 2 will probably be about 15 filling in the blanks, using that page that y'all just practiced with, and using those two sites on visible body. Fair?
We have a word bank.
No. And spelling will count. Spelling will count. So you were trying to be phonetically correct, because, I mean, you'll get some points, right? So don't leave blanks. Don't leave blanks. Always guess or something. Okay, so that's the lab. I am going to go to my office. I would really just love to meet with some of you who I think, if you think that I could offer any kind of whatever, but I would really like to meet with the ones that haven't passed anything yet. And you kind of know who you are. So, because I really just want to help. I want to help. All right, and this is a condensed class, so we have to do 16 chapters in 8 weeks. By the way, this first part, let me say this before everybody's making too much noise. This first part I spent a lot of time on, because I think this is usually the part people are the weakest. When we get to the organ systems, I roll. Because at that point, you all should be knowing what you're going to need to do in here, and I am rolling on that. Okay? So let me help you if I can.